Hi, everybody. Welcome to Micro Seminar. I'm Lizzie Wilbanks, and it's my pleasure here to introduce Wil Leavitt today, who is going to be our speaker for Micro Seminar. Wil is an assistant professor at Dartmouth College in the Department of Earth Sciences and with an adjunct appointment in biology as well. And Wil is going to tell us all about sulfate-reducing metabolism, taking vital signs after death. And I am really excited to catch up with Wil about what he's doing. He does great work on sulfur cycling geochemistry and biology, really kind of bringing the two together at the interface. So with that, I will leave it to Wil. Take it away.
Awesome. Thanks so much, Lizzie. Let me find out how to share my screen real quick here. Yeah, so actually, before I flip over to my talk, I just wanted to say a big thanks to all the Micro Seminar folks, Lizzie for inviting me, Cameron Thrash, and David Baltras for helping us get organized, and then also to Jen Biddle, who founded this. This has been a really fantastic opportunity out there for a lot of my colleagues I know, and I was really excited to get the invite. So thank you so much. It's already led to a few collaborations from other folks' talks. So with that, hopefully it doesn't do the infinity screen thing. Okay, and there. So actually, Lizzie, if you could just confirm that you see my slides.
Yeah, they
Awesome.
yeah, they look good.
Thank you. Okay, so regular talk introduction. I obviously thanks to the organizers, and then I have to say thanks to all of my many collaborators, from my PhD advisor, Dave Johnston, through my postdoc advisor, Alex Bradley, and the folks in his group, and then all of my various collaborators over some of the recent projects that we've been working on through my postdoc, and some of those that I've brought here to Dartmouth as well. And especially a big thanks to NASA, who's funded the last few years of all of this research, and then to the ACS, who's funding some of our ongoing research. So I like to take a huge step back, and this is really where I like to start to frame sort of the way I think about microbial geochemistry and geomicrobiology. And this is an image from Voyager, looking back at the Earth about 4 billion miles away. You can see Earth just as this little pale blue dot, and it's taking up only about 0.12 pixels of this entire image. And the only reason it lights up is there's a little band of light from the sun that it's shining through. And I love images from NASA like this, because this is really helps me sort of think about the big questions that I get excited about on a daily basis, or hopefully at least on a weekly basis if I'm thinking big, even when I'm sort of stuck in the lab thinking about the smallest little details of sulfate reducers. And these include, how did microbial life originate on Earth, and how do life and microorganisms interact? And then, of course, thinking beyond Earth is how widespread is life in the universe, and what can microorganisms on Earth tell us about where we could, in theory, detect life elsewhere in the universe, and what might that life actually be like? To put this into a little bit more of a terrestrial context or an Earth-based context, I think one of the biggest challenges as a microbiologist and as a geochemist is thinking about how do the actions or the interactions of microorganisms on incredibly short time and spatial scales influence the Earth, and over the course of its evolution over the last 4.5 billion years or so. And then vice versa, how do these changes on Earth influence the origination and evolution of different microbial metabolisms? What are the forcings and feedbacks? And so I like to think of the Earth as essentially just a big bucket of chemicals, and life and microorganisms as just little catalysts. But really, it's the interaction. This is where really the interesting questions and observations I think arise. And so with this sort of context is where I like to dive in to thinking about how do organisms and microorganisms in particular generate geostable signatures or geochemical signatures. And so you can just take your sort of generic microbial metabolism or microbial cell, and you have a bunch of forcings from the environment, things like temperature, pH, concentrations of electron donors and acceptors, of other various micronutrients. These influence the organism's ability to grow, the various types of protein expression it can do, the ratios of protein expression, the general redox state of the cell in reference to the environmental redox state. And then, of course, things like the metabolic concentrations within the organisms themselves or within the populations. And then we can ask, what are the outputs that are going to be potentially preserved in natural systems for short and potentially longer timescales? And when I say short, I mean, weeks to days to hours, to longer time scales, to millions and even billions of years potentially in the geological record. And so these are things like lipid structures, potentially the isotopic composition of those various lipid structures, so the carbon, nitrogen, hydrogen isotopic composition of those, as well as the isotopic composition of various inorganic products and reactants that these organisms interact with. So carbon, oxygen, sulfur, I should put nitrogen in there, as well as the various types of mineral phases that can actually be produced or are produced as a result of microbial metabolism under the influence of different sort of redox or net metabolic state. So these I like to think of as essentially your very overly simplified box model of a microorganism or a microbial population. Putting this in a little bit more of the geochemical context, I think it's really useful to recall that all domains of life can do essentially the easy metabolism, as I call it, aerobic respiration, as we see all the way over here on the left. But it's really only the bacteria and the archaea, the microorganisms, as I'm generalizing them here today, that can perform anaerobic respiration or anaerobic oxidation of organic matter. And these include respiratory pathways such as sulfate reduction, which we're going to talk mostly about today, iron reduction, CO2 reduction or methanogenesis. But of course, there's a whole host of other anaerobic metabolisms that only bacteria and archaea are capable of. But it's really critical when you're thinking about carbon cycling more generally, as these are the ways that organic matter is being incinerated in anoxic systems on Earth. And these are most likely the abundance of metabolisms on Earth prior to the rise of oxygen, approximately 2.5 billion years ago. So really, if we want to understand this origin and evolution of life on Earth and potentially elsewhere, we really need to focus in on these anoxic microbial metabolisms. To put this in a little bit more of a quantitative context, there's a fun calculation you can do where you think about how much oxidant is in these three different reservoirs of oxygen in the atmosphere in today's abundances, sulfate as it is in seawater, and iron oxides. And what you can calculate, sort of back of the envelope from this Hayes and Waldbauer paper, is that you get about 37 times 10 to the 18 moles of oxidant in oxygen. So this is 37 examoles. You get about 13 times that in sulfate if you calculate the amount of oxidant and all that sulfate in the oceans. You actually get about 50 times that in iron oxides. But as I always like to point out, it's really important to think about where your oxidant is available. So oxygen is relatively insoluble. You can only get about 200 micromolar in surface seawater these days. Whereas sulfate, you have about 28 millimolar in the oceans. And of course, there's some in lakes and freshwater as well. And iron oxides, while it's readily abundant, it's primarily insoluble at Earth's surface temperature and pH conditions. And so while there's a lot of iron oxide out there, it's a challenging oxidant to access. And I know from some of the previous iron micro seminars, we think about the really amazing mechanisms by which organisms perform iron reduction. But sulfate reduction in many ways is sort of the optimal, I like to think, strategy if you can't use oxygen, but you need a readily available oxidant. And so it's this sort of large-scale observation has led one of my heroes, John Hayes, to make the observation that oxygen is merely the icing on a ferric sulfate cake. And I think this is really encapsulates that sort of time and space component that I always like to frame my work in, is that, you know, over the course of the last 4+ billion years of Earth history, oxygen's been a minor player, and it's really the cycling of these other oxidants, sulfate, iron oxides, CO2, nitrate, nitrite, all these other intermediates that have probably been major players in the cycling and recycling of organic matter on Earth. So with that, we're obviously going to focus in on sulfate reduction today, because that's what we work on. But I like to use that as essentially my setup for why bother with sulfate reducers. So now let's dive into where we think they might matter. So this I love is sort of a nice quantitative pictogram. I'm sure Marshall would call it something else from this Bulls et al. Paper. But I like to use this as a way to sort of frame the fact that you get sulfate reduction at various rates over a range of about 10+ orders of magnitude in rate in the modern ocean. And so you can sort of see that over this range in color in this artificial neural network plot that Marshall has generated. To sort of oversimplify it, which is what I like to do wearing my geochemist hat, we can think of sulfate reduction as occurring primarily in anoxic marine sediments, although it of course occurs in terrestrial systems and soils as well. But quantitatively, we think of sulfate reduction as being a big player in the remineralization of organic matter in anaerobic sediments. It's calculated that anywhere on the order of 30 to 70%, so I just take an average 50% here, of organic matter that reaches the sea floor is actually remineralized via sulfate reduction, generating hydrogen sulfide and DIC or CO2. But actually in the vast majority of that sulfide is reoxidized, somewhere on the order of about 90%. And so those electrons are dumped onto other oxidants, such as those iron oxides or oxygen itself. And actually an extension of that little back of the envelope calculation that Hayes and Waldbauer performed is that you can think of one in every 5 breaths that we take of oxygen has somehow cycled through this sort of sulfate reduction reoxidation cycle at some point, given the way you calculate it. But I just like to use this as a way to frame that, sulfate reduction, while it might be constrained to various anoxic systems, it has a direct impact on the relative abundance of oxygen in the atmosphere today. So today we're going to talk almost exclusively about sulfate reducers, and specifically the respiration, or the anaerobic respiration pathway of dissimilatory sulfate reduction. It's only performed by microorganisms, and specifically I mean by primarily bacteria, though there are some archaeal pathways of this. And as geochemists, we like to generalize this as just the reduction of sulfate using 8 electrons from organic matter or hydrogen to produce hydrogen sulfide and CO2. So this is a nice simple way to write out that reaction. Now, if we want to track this process in time, and especially in deep time, we have to come up with a way to trace that. And this is where stable isotopes really come into play, and natural abundant stable isotopes specifically. And so for today, I'm going to try to keep it very general in terms of the sulfur and then the hydrogen isotope notation that I'll use. So for sulfur, we always think of the lighter isotope of sulfate, or of sulfur, S32 in this case, reacting slightly faster than the heavier isotope of S34. And so the more times you do this reaction of sulfate reduction, the more likely you are to accumulate product that's light. But this differentiation between the heavy and the light isotope in this case is directly dependent on the net rate of the forward reaction. And so this is primarily a kinetic isotope effect that we're going to talk about today, although we'll mention equilibrium a little bit. So the notation we're going to use is standard delta notation, delta S34, and we're just going to report isotopic fractionation in units of per mil, or parts per thousand. And so when we report a deviation in the relative ratio of S34 to S32 in a sample versus a standard, this is the per mil notation that we're going to be using. And this notation is exactly the same for hydrogen isotopes. You can just swap S32 for hydrogen and S34 for deuterium when we switch over to lipids in a few minutes. That said, so to back up to sulfate reduction, that bigger sort of sulfate to sulfide fueled by organic matter reaction, that's actually just a simplification of a whole bunch of enzymatic steps that we as microbiologists actually like to think about in more detail. And understanding the biochemistry can actually inform our understanding of the isotopic fractionation that we're going to talk about in a few moments. And so we'll get into the details of sulfate reduction in a little bit more gory detail, but generally we think of sulfate as being transported into the cell, being activated to some high energy intermediate known as APS, but we haven't done any redox chemistry yet. At this point, this is where that APS can be reduced to sulfite, SO3, by this enzyme APS reductase. And then at that point, you've only done 2 electron transfers. And so from sulfite all the way to hydrogen sulfide, this is where a lot of the energy is going to be conserved. And so the coupling of this set of electron transfers to the inner membrane, this was actually sort of a mystery up until about a year or 2 ago, 2 years ago, when this paper by Santosh et al. Came out in Science. And it's a really elegant paper sort of detailing the biochemical mechanism of energy conservation, coupling the reduction of sulfite ultimately to hydrogen sulfide. So I'll just leave it there for now, but this is where this intermediary carrier DSRC comes into play. But so actually, let me back up a little bit. So this is where we think a lot of the enzyme isotope, or a lot of the isotope effects are occurring, is at these reductive intracellular steps. So the reduction of APS to sulfite, and then the reduction of sulfite to these intermediates, and then the reduction of those intermediates to hydrogen sulfide. And so if we want to understand the larger geological and geochemical context, we need to really dive into the biochemistry. So this is what I like to think of as the catabolic reactions of sulfate reduction. But there's also the anabolic reactions of sulfate reduction. And this is where I started to jump into trying to understand the hydrogen isotopic composition of biomass, and in this case, specifically the lipids. And we'll come back around to this in a few minutes, but essentially I like to think of the sulfur isotope signatures as being a very catabolic or net metabolic, energetic state, relating to the net energetic state of the organism. whereas the hydrogen isotopic fingerprints relate to the net anabolic set of reactions. And so to motivate the thinking about these two different systems, interpreting sulfur isotopes over the geological record is really where we go when we're applying these sulfur isotopic compositions. However, in thinking about in other systems as well, in modern estuarine sediments, you could apply these just as well. And then thinking about using hydrogen isotopes in lipid biomarkers as a proxy for essentially what metabolism is operating, or the way I'm starting to think about it is how effectively or how efficiently is that metabolism operating. So first we'll talk about sulfur isotopes through Earth history, and actually then dig into some of the proteomics there, and how we can hopefully reinterpret sulfur isotopic records through the proteomic lens. And then for the last just few minutes of my talk, I'll discuss a little bit of the hydrogen isotope work that we've been taking off on. Excuse me. So I guess the bigger motivating question behind all of this is what bioactivity metrics of metabolism exist or might exist in the sedimentary rock record or the soft sedimentary record in modern systems. And so this is sort of every sulfur isotope measurement up through a few years ago of a rock. And so sedimentary sulfides in blue, or sedimentary sulfates in red. And basically sort of the first order observation I like to point out on this, Earth history, all of Earth history on the x-axis versus this range in sulfur isotopic composition of these two different pools, is that in the Archean, or the first age of Earth, we have essentially not a whole lot of fractionation or compositional change in sedimentary sulfides. We go through some interval known as the great, this camera just kicked off. Oh, there we go. Okay, sorry about that. We go through some interval of known as the Great Oxidation Event about 2.5 billion years ago, and then we start to see this expansion in the isotopic composition. And then as we move forward in time, we get a further expansion in the oxidant reservoir, and potentially a larger and larger range in fractionation. And what this has been attributed to in the past is essentially this increase in the difference between the sulfate composition, the red, and the sulfide composition in blue, or as we're summarizing it here, an epsilon value. So this is just the difference of two different delta values. The increase in this epsilon value through time is somehow related directly or indirectly to the abundance of oxygen or oxidants in the Earth system. And so the sort of the simpler or the earlier interpretation of this record is that in the Archean, you have a low concentration of sulfate in the oceans. Through the Proterozoic, you have somewhere between the Archean and the modern value of about 28 millimolar. And then through the last 550 million years or so, we get somewhere on the order of the modern concentration of sulfate, but we don't have really a good quantitative constraint on where. So you essentially get these order of magnitude shifts from the Archean to up through the modern in the abundance of that sulfate in the ocean. Now, one of the challenges with this interpretation is that this assumes that sulfate reducers are always operating exactly the same way and always at exactly the same rate. And so remember, we're talking about a kinetic isotope effect today. And so rate is really what is, in theory, one of the major players in this. And so this is this big assumption that goes into this interpretation. And it might be correct, but it's one worth testing. And so this is something that we spent a few years thinking about and working on during my dissertation. One of the key observations I think that challenges this assumption is that sulfate-reducing microorganisms can produce the entire range in delta values, so in this per mil value, or the fractionation between sulfate and sulfide that we see in the geological record. So pure cultures of sulfate reducers in my laboratory or in a bottle in the laboratory can produce this entire range. And so this really challenges us to think more deeply about what does this range in isotopic fractionation mean, not just at the environmental level or the geochemical level, but actually at the microbiological and the biochemical level. And so sort of summarizing 60 years of experimental data in one histogram or in one box and whisker plot, we can see that on the order of about 700 experiments, we see this entire range in the geological record summarized and can be done just by microbial sulfate reducers in pure culture. And we'll expand on this a little bit more in just a moment here. So I wanted to understand what controls this range in fractionation with just these pure cultures, and what more can we actually interpret from these environmental values or potentially even these rock record values by understanding what controls the microorganisms. And so to do this, we actually set up a number of chemostats or continuous culture experiments. And so in these systems, you essentially have your culture held at steady state, or a chemical environment and static as a chemostat is. And we're actually going to limit these organisms either by electron donor or by sulfate concentration. But for the next few experiments, we're just going to limit them by electron donor, and then we're going to deliver that electron donor over a range in rates, but get those up to a steady state. And then the nice thing about this type of experiments is you can measure the isotopic composition of the sulfate and sulfide that come out the back end of your reactor, and you can just take the direct difference of those, and that's your isotopic fractionation factor. And so basically when we do that over a whole range in sulfate reduction rate here, just as calculated as a volumetric flux, we can observe this huge range in sulfur isotopic composition here on the y-axis simply as a function of how fast I feed my bugs. One interesting thing I like to point out on this plot is that, so we see this really interesting nonlinear inverse trend, and we'll come back to this in a moment. But there's this interesting minima that we were able to get in our chemostats, always in and around 17 per mil, which I'm not going to talk too much about this part of the story today. But when we actually went in and measured the isotopic fractionation of one enzyme, the dissimilatory sulfite reductase, that rang in at about 15 per mil. And so if there's a consistency between these sort of whole in vitro and in vivo stories, it's that the sulfite reductase itself is actually the rate limiting step at these faster rates. Now, we'll need to measure all of the enzyme-specific isotope effects to truly test that. But I think for now, that's essentially our working hypothesis. But now what we can ask is, do these experiments, do these lab-based pure culture experiments tell us anything about what's going on out in natural systems? And before I jump to that, let me remind myself that basically what this also tells us is that most of the literature is very biased towards the fast rate end member. We can see here is that the bulk of those 700 experimental data points fall at the lower fractionation values, and this really tells us that we're doing our experiments a little bit too quickly in the lab to really replicate these higher fractionation values. But if we jump to essentially a very small compilation I was able to pull together from Kaplan and Rittenberg, Goldhaber and Kaplan, excuse me, we can actually see that out in natural systems where we get much lower net sulfate reduction rates. The bulk of the data in those natural sediments, marine sediments, tends to cluster towards the slower rate end member and the higher fractionation values. Interestingly, though it's poorly supported by the few data points we have, the trend, the minima is the same, but the function that we can actually put through this data, this nonlinear inverse trend, is exactly the same function. And so at this point, it's just kind of an interesting consistency argument, but we're hoping to go in and test this actually in more modern marine sediments where we can actually get direct rate measurements, so using something like the radiotracer method, as well as getting natural abundance stable sulfur isotope measurements from either the same or parallel cores. But I think for now, the takeaway from these chemostat experiments is that we can actually do a pretty good job of explaining modern marine sulfur isotopic signatures in systems where you're getting relatively slow sulfate reduction rates, though we're obviously sort of missing a little bit of the overlap data in both our lab organisms as well as then faster rate end members in our experimental systems. But to dig into this a little bit more, we wanted to actually try to think about, okay, can we actually apply a metabolic model, or I should say an enzymatically constrained model, to try to interpret this laboratory data and then potentially reapply that to reinterpret this environmental data. Now, there's a couple of different ways you can do this, and this is I call it the chaos slide, because basically this is just sort of a distillation of using a Michaelis-Menten-Monod relationship to relate the net fractionation to something like the environmental abundance of sulfate, as well as the organism's ability to take up that sulfate, and then the net reaction rate, so the cell-specific sulfate reduction rate, as well as some activity coefficients. And basically what it comes down to, and as I like to say, don't worry about the equations for now, you can read the paper if you really want to, it comes down to a relative abundance and activity, affinity for the sulfite, or the sulfate and sulfite, and then the electron donor. What's really neat about this simple model that my colleague Alex built to explain a lot of this chemostat data is that you can basically see that over now just a relative range in fractionation or a relative sulfate reduction rate on the x-axis up on top, excuse me, is we can basically reproduce those nonlinear inverse trends very cleanly, where concentration is essentially pushing your rays up and down, but over a very narrow range. Alternatively, you can ask the inverse of this question is, how does sulfate concentration really affect the range in fractionation. And again, on just a relative axis of fractionation. So you can think of 0 as being, again, 0, 1 as being that maximum observable fractionation of about 60 to 70 per mil. Basically, you get sulfate concentration is just sort of tamping down the maximum amount of fractionation possible. And so you can sort of have these two different end member controls, or you can have some mixture of the two. But really, it comes down to this availability of the electron acceptor relative to the electronic donor. I always have to put in a really good pitch for my colleagues, Itai Halevi and Baz Wing, and their postdoc, Christine Wenck. They actually modeled this in a completely independent way. Thankfully, they modeled my data, so or our data, I should say. And so it's nice that we have two totally independent modeling approaches. And what's interesting about this approach, where they use this force flux model, and again, you can go read about the details of it, that sort of distills out in this one equation here, is that they're able to tune their model based on intracellular metabolite and protein concentrations. And so they're able to reproduce this same nonlinear inverse trend as a function of rate versus fractionation. Oops, I cut off my axis there. That's fractionation on the y-axis. And so this is nice because now we can actually go back and we can test their predictions that they put into their models, or the parameters they put into their models, and we can actually go test them experimentally. And so that's actually what we've been able to do with some of the proteomic work. We're now working with our chemostats. So here's a picture of the little chaos chemostats, as I like to call them. These are the ones I built during my postdoc here on the right, operating in parallel now. Here in my lab, we've actually gone to commercial systems, in part just to save us the headaches of building them ourselves, although they have their own headaches, of course. But so basically, we wanted to repeat some of the earlier work that we'd done, where we saw lots of these, the little red data points here that I've just replotted. And we just wanted to pick essentially two rate end members. And so we picked essentially what I'm going to call a fast rate end member, and then we picked a slow rate end member, and that should actually be down here at about 20 per mil. So we picked about a 10 per mil and a 20 per mil rate end members, and we run replicate chemostats, so two chemostats running at the fast rate, and then we spin them up to steady state. We harvest sulfur for sulfate and sulfide, sulfur isotopes, and we actually harvest biomass as well in this case to look at the intracellular abundance of the various proteins, or excuse me, the relative abundance of the various proteins in these replicated chemostats. And then we'll do that over about 4 or 5 different independent samplings. So we have really high replication, both biologically and technically, at the fast rate end member. And then we do exactly the same thing at the slow rate end member in a separate experiment. So what's really neat about this is that we can actually, working with Jake Waldbauer at University of Chicago, we can actually get relatively quantitative proteomic measurements in these two different conditions, and we can compare them directly and ask, essentially, which parts of the metabolism, or excuse me, which of the enzymes are up expressed or down expressed. And so any up expressed value that I show you in the next few plots, that's essentially the fast rate end member. Oop, there's my arrow. And anything that's negative or down expressed, that's actually up expressed in the slow rate end member. So it's a little confusing at first, but you can basically think of it as positive as fast, negative as slow. This is just a quick plug for Jake Waldbauer's recent paper. I think it has a deal or a number now, but this is the proof from about 3 weeks ago. So if you're excited about proteomics, and especially quantitative proteomics, please check out this paper. If it's up, or when it's up, I'll try to post it or tweet it to MicroSeminar. But this is the way he does this. Essentially, gives us really the ability to look for relatively small changes, even sub log 2 abundant changes in an expression. And so this is really powerful if you're looking for subtle but really important shifts in the proteome. So this is just a quick snapshot of those fast versus slow experiments. And so everything on the right half of the plot is essentially increasing in expression in abundance in the fast chemostats. Everything to the left of the 0 is up expressed and increasingly expressed in the slow relative to the fast. Everything in yellow is statistically significant given a very harsh or a very stringent Bonferroni cutoff, and everything in blue and red is essentially statistically insignificant. But you get and we'll dig into a little bit of the details here in terms of the sulfate reduction pathway. But at this point, we're still sort of parsing through this data because there's so much information in here in terms of the different parts of the sulfate reduction metabolism and then the whole organism's metabolism that are changing. And so actually, one of my colleagues, Sophia Venceslav, one of the biochemists I work with, has done a nice quick analysis for us to sort of bin these. And so you can see that energy production and conservation. So blue is the fast chemostats, red is the slow chemostats. you can see a little bit of a change in the number of proteins that are statistically significantly increased or decreased. Not surprisingly, you see a massive change in translation and ribosomal structure and biogenesis. The fast organisms are dividing faster. And then one of the interesting, and as usual with microorganisms, we see function unknown, a big change in the slow-growing organisms relative to the fast, and then signal transduction actually, which is interesting. And then of course there's a number of other potential stories in this data as well that we're still digging into at this point. But to focus in a little bit on just the sulfate reduction pathway enzymes themselves, so sort of summarized in that same type of plot up on the right, but just here on this sort of more simple plot, so we can see that sulfate activating enzyme, that APS, or that sulfate APS reducing enzyme, we see a significant increase in the relative abundance of that sulfate, that's APS reducing enzyme, as well as the QMO complex, which is actually, that donates electrons ultimately to that APS reductase. The sulfite reductase, the DSRAB, we see a little bit of an increase, or sorry, yeah, that's the other subunit, the SAT, and this is the sulfate acquisition machinery. The DSRAB, or the DSRA here, we see statistically no difference. And so this is actually really interesting when we plug this back into our bigger, sulfate reducer metabolic and isotopic model, and that we don't see any difference in this sort of core sulfate reduction enzyme or sulfite reduction enzyme. And then interestingly, in the slow organisms, we see a significant increase in the relative abundance of the lactate dehydrogenase, so the electron acquisition machinery, as well as these RNF, or these hydrogenase complexes. And so this is just a quick snapshot of a lot of that data for these 12-hour turning over organisms relative to these slow or 3-day turning over organisms. But this sort of gives you a taste for the types of data that we're able to get out of these types of experiments. And then hopefully we'll be able to plug these back into our sort of predictive metabolic isotope models. Yeah, and so just to sort of simplify or summarize this simplified view is, in the fast-growing organisms, we see this sort of uptick in the sulfate acquisition and activation machinery. We don't really see a huge change, or I should say we see no change in the sulfite reduction machinery. But in the slow-growing organisms, we see a big uptick in what I would like to call the electron acquisition machinery. And so this is sort of interesting when you start to think about this, the fast-growing organisms. We're providing them a fair amount of sulfate, but they're still electron donor limited relative to sulfate, but they're still bringing in lots of sulfate, it seems like, relative to those organisms that are electron donor starved and forced to grow quite slowly. They're really cranking up their ability to acquire, or I should say, use the electron donor. So this is just putting that same set of observations back on essentially a simplified metabolic map of the sulfate reducers here. So the increase in expression in red there. But when we have to think about this in a much broader context, and this is what my biochemical colleagues like to plot this data. And for me, I always get a little panicked when I look at these types of diagrams. You kind of really have to walk through them slowly. But again, we have essentially those enzymes that are increased in the fast-growing organisms. I flipped my color scheme, I apologize, in red here, and then those enzymes that are down expressed or down and fast relative to slow in blue. So the FLOX HDR, dehydrogenases, and then again that RNF complex. But we're going to basically be able to go through and do this in a quantitative way for essentially every enzyme and every part of the pathway that we understand, as well as hopefully those parts of the pathway that we don't understand, and generate some new predictions and some new hypotheses about what are these, what are the relative importance of these different enzymes in producing isotope effects, and then flipping it back on its head, what are some of the important enzymes under these sort of energetic starvation states for these organisms. To switch gears a little bit, but still staying intracellularly, there's a nice set of experiments that our colleague Minsub Sim did and just recently published in GCA, where over, in this case, a batch experiment, he was actually able to measure the intracellular composition and concentration of different important sulfur pools like the sulfate, the APS, the sulfite, and the sulfide. And what's really neat about this method that he developed is that basically he's able to sort of test some of the predictions put out by these metabolic models for the isotopic composition of these different pools. And so sort of integrating some of the types of predictions we make using the models, and then going in and testing them, not just at a proteomic or a metabolomic level, but at an actual isotopic composition level. And so I encourage you to go check this paper out if you're interested. But one of the I think one of the coolest points for me is that the intracellular composition of sulfate here in the 6-pointed stars, those are incredibly enriched. Oh, excuse me, switch slides. Those are incredibly enriched here on an axis of delta S34 in his batch incubations, where you see an increase in the growth of the cells, so the triangles. Your net extracellular sulfur isotopic composition of the sulfate and the sulfide, you're getting a very tiny difference of only a few per mil, but the intracellular sulfur isotopic composition is massively enriched. And so this really is interesting in that even though they're expressing a very tiny isotopic fractionation between net external sulfate and sulfide, the intracellular composition in these presumably fast-growing cells is really enriched. And so applying essentially what I call a dirtier version of MinSub's technique, although he actually made the measurements, I just didn't do the preservations quite as well. We can actually do this in our chemostats, or we tried to, I should say. And so we can look at on a log axis of the abundance of our sulfur pools, so the extracellular or the incoming sulfate in our fast versus slow reactors in red versus blue, the residual amount of sulfate left over in these reactors, again in red versus blue, and then the cellular sulfate that we're actually able to extract from just the biomass itself. We see a relatively large difference in the abundance of sulfate in the fast-growing cells relative to the slow-growing cells. And this is a consistent story for intracellular APS and intracellular sulfite. Now, we're only able, in our case, to get sulfur isotopic compositions for the intracellular sulfate. But let's start with here's the incoming sulfate-sulfur isotopic composition, just on a delta axis. And you can see that in the reactor sulfate, consistent with what we would predict, the fast-growing organisms don't fractionate as much, so that's the difference between the sulfate composition here and the outflowing sulfide composition. And then the slow-growing organisms fractionate a bit more in the incoming, the residual sulfate and the residual sulfide. And what's really interesting is when you compare the two reactors of the fast-growing organisms, so red, so intracellular sulfate, versus reactor sulfate, you see a pretty big shift in the red of the residual sulfate versus what's in the cell, relative to the slow-growing organisms where there's the residual sulfate in the reactor in the experiment versus what's found in the cell. And so this is, if anything, this is consistent with what MinSub's observations are finding. And so this is really interesting in that, again, we can now couple these sort of intracellular measurement, or these measurements of protein expression with this sort of the quantitation, of the intracellular pools of these different key metabolites, as well as potentially their isotopic composition. This kind of brings me back to that paper by Wenk, Wing, and Halevi, in that they make predictions for what some of the other intracellular metabolites ought to be under different, essentially, growth regimes. And while we don't have these same intracellular metabolites measured in these experiments, in their reduced to oxidized ratio, what is really interesting is that they predict a relative utilization of things like ferredoxin when the organisms are growing really fast and therefore fractionating not quite as much, but a big shift in which intermediates or electron carriers are being used. And while they require these different changes in these different pools to essentially transit this y-axis in isotope space, we can actually go in and test these predictions in our same types of experiments. And so that's something we're actually hoping to do by reanalyzing some of the proteomic data to see if we get changes in, say, the ferredoxin cycling enzymes or the minoquinone cycling enzymes. But we're also going to hope to go back in and do these same types of experiments at sort of the high fractionation, low rate, low fractionation, high rate end members, and actually look at some of these intermediates and see if we can look at their relative abundance as well as potentially their redox state. So this is, I think, really where we're going forward and digging a little bit deeper into the understanding of the metabolism and some of the metabolomics is going to potentially help us to, again, reinterpret this environmental data and this whole cell data that we have. So where does this leave us for sort of this first, 2/3 of the talk here? We get this predictions coming from some of these metabolic models that enzyme abundances don't necessarily scale with rate. But what our experiments show us is that some enzymes actually do scale, their abundance does scale with rate. However, things like the DSRAB, for example, they didn't change abundance as a function of this change in rate. But either way, these are really powerful constraints when we take these quantitative proteomic measurements and apply them to our sort of predictive, or we use them to calibrate our predictive isotope fractionation models at this sort of the whole organism and sulfate reduction pathway scale. So that sort of wraps up the first part of, or I should say really the first 2/3 of the talk. I'm just going to speak a few more minutes about hydrogen isotopes. And it's sort of a relatively, a little bit of a quicker clip, so I'll leave out some detail. But I think it's really these combined approaches of sort of the modern quantitative proteomics and sort of classic stable isotope geochemistry, but applied at a new level in terms of precision and scale is what's going to help us understand not only how our sulfate reducers operating in the lab, which is always easier said than done, but how are they operating in natural systems where we really want to interpret these isotopic signatures. And so to do this, I think we're in need of essentially other bioactivity metrics. Now, direct rate measurements are always optimal, I would say, in that if we can go in and directly measure the rate and the abundance of different organisms and different metabolisms, this is ideal. But you can't do this when the organisms are already dead. And so this is really where I think it's important to try to understand what are the other sort of markers of how active and how metabolically charged were organisms after they're dead, and what are these potential proxies. And so this is where I think lipid abundances, lipid structures, and then of course the hydrogen isotopic composition on those lipids can potentially be really powerful going forward, and then combining it with these other proxies, the sulfur isotopes, the proteomics, et cetera. And so this is really all piggybacking on the work that's come out of Alex Sessions's group, Dave Valentine's group, and Victoria Orphan's group in Santa Barbara and Caltech over the last few years, and many of their students and postdocs. Here just highlighting one piece of work from Maggie Osborne, but seeing that on an axis of essentially hydrogen isotopes in the lipids, so this is thinking about the relative abundance of deuterium to hydrogen, just the way we were thinking about the relative abundance of S34 to 32. In this case, we're just thinking about those hydrogen isotopes on things like fatty acids and their lipid structures and different lipid structures. But so on an axis of epsilon 2 in this case, still in units of per mil, we can see that there's this huge range in natural abundance isotope fractionation. And that sulfate reducers themselves actually have a relatively large range of a few hundred per mil in fractionation between the lipid hydrogen isotopes and the water that these organisms are actually growing in. And the question for me is really, what if anything is this telling us? And the prediction, I should say more than the question, is that it's telling us something about metabolic state and really the net redox state of these organisms. And so this is essentially sort of unpacking some of that data from that Osborne paper. And so you can see aerobic heterotrophs like E. coli or denitrifiers show this relatively large range in isotope fractionation between lipid and water, depending on who it is and how you grow them. And similarly for the sulfate reducers, you get this depending on who you're growing and then how you're growing them, meaning which the combination of electron donors and sulfate you're growing them on, you get this huge range in hydrogen isotope space between that lipid and water. And so really this motivated me to ask again the question of mechanistically, what is driving this range in fractionation? And if we can come to understand this sort of the signal here in all of this rich data, and really understand it at the biochemical level, this potentially is going to allow us to go out to natural systems and really interpret robustly hydrogen isotopic signals in lipids. And so this is a nice figure by my colleague Sebastian Kopp that's essentially summarizing that same work from the Sessions lab in a nice op-ed, or not op-ed, I forget what they're called, perspective in PNAS from about 8 years ago now by Dave Valentine, is that lipid hydrogen isotopes are essentially just a compositional mixture of various pools. But one of the really critical pools intracellularly is the hydride, or the hydrogen coming off of the NADPH, and then its isotopic composition potentially has a really large impact on the hydrogen isotopic composition of those lipids relative to the water that these organisms are growing in. And then the way you make your NADPH and how much you make, depending on if you're a hydroautotroph, as he's calling them here, a hydroheterotroph, that can actually dictate the composition of your lipids. Now, to put this into some context with sulfate reducers and many other organisms, one of the key enzymes that plays a role in this process is known as transhydrogenase. And the reason I'm just going to focus in on this enzyme in particular for the next few moments here before I wrap up is that this enzyme probably has one of the largest stable isotope fractionations in nature, on the order of 800 plus per mil. And this enzyme is basically taking the hydride off of NADPH and flipping it onto NAD and vice versa. And so this is really a way you're translating that hydride between essentially the biosynthetic, the catabolic, and the anabolic pools inside these cells. And then the scales of these pools are going to probably scale with how actively dividing versus respiring these organisms are. And just to name a few of the transhydrogenases that are out there is the PNTAB, the NFNAB, and the UDHA. We're going to focus in on the NFNAB, or the electron bifurcating transhydrogenase in sulfate reducers for the next few minutes here. And so it's this large isotope fractionation factor predicted, or that's been measured in vitro, that is the reason that we're going to focus in on this potential important step in lipid hydrogen isotopes in sulfate reducers. And so you can sort of set out a couple different hypotheses, or a couple of versions of the same hypothesis, is that if we see large swings in fractionation between lipid and water, are these changes in the step that's actually fractionating during hydride transfer, or is this actually just sort of a cellular scale distillation effect of that hydrogen isotopic composition? And so we sort of set out to test that in our sulfate reducers. I'm going to plow ahead into the sulfate reducer data in just a second here, but this is really one of the most beautiful experiments that came out of my postdoc advisor, Alex Bradley's lab, done by him and his lab manager, Melanie Cease, is that they basically took a wild-type methylobacterium and then a parent mutant where the transhydrogenase had been kicked out, and then they put in a foreign pathway. And then it was evolved. This is actually done in Chris Marks's lab. It was evolved for 3, 6, and 900 generations, I believe. And what you actually see is you see an increase in the transhydrogenase expression. And when you get that increase in transhydrogenase expression, you get a dampening of that isotope effect, which is really interesting because this then makes predictions for the relative abundances of these different hydride-bearing pools, these NADP and NAD pools, and who has the hydride and how much of each of them are there in the cell. These are relatively challenging pools to measure, but I think this is essentially the next step in this type of experiment. And so in a little bit of a less beautiful way, I wanted to do a similar type of experiment where we actually influence the sulfate reducer and see what the outcome is in terms of hydrogen isotopic composition. So basically, the question is, again, does transhydrogenase play a role in determining the lipid hydrogen isotopic composition, the epsilon-2 lipid water in sulfate reducers? And so I'm going to cut to the chase here and basically show you that we can basically knock out that transhydrogenase, or borrow transposon mutants from people that know how to do that stuff, from the Berkeley group of Adam Deutschbauer. And you can grow them in different ways to force them to grow with pyruvate or with malate. And basically, when you have the transhydrogenase knocked out and you grow them on malate, they do a really poor job. They grow really slowly relative to the wild type or relative to themselves when you grow them on pyruvate. So you get this really nice phenotype. And basically, we then can go in and then analyze the individual fatty acids, which is what each of these dots represent, and then average that, take a weighted mean of that data, and that's what the bar is. And so essentially, these are individual fatty acid values. This is the weighted mean of those. And you can measure the hydrogen isotopic composition of all of those different lipids in the wild type and in the mutants grown under these different conditions across multiple biological replicates. And so basically, the punchline of this is that the wild type and the mutants really don't change their hydrogen isotopic composition either at the individual lipid level or at the weighted mean level when you grow them on pyruvate. But when you grow them on malate and they don't have the transhydrogenase, they basically fractionate less and less and less. So in this case, they don't have this transhydrogenase active. They're growing on malate, and they fractionate significantly less when they're forced to grow on malate. And so the prediction here is that we're basically influenced the intracellular and NADPH to NADP ratios, and this is having a direct effect on that cellular scale and individual lipid scale isotopic composition. And so now at this point, we're probably not going to pursue this at the moment, but I think in Minsub Sim's group, they're actually going after these intracellular metabolites and trying to quantify them really robustly. And so we can do this over a range of different electron donors and even grow them as fermenters. But to cut to the chase, what's really interesting is that you see, again, as a function of growth rate, you get this nonlinear inverse trend in, again, the weighted mean of the fatty acid isotopic composition relative to the water that they're growing in. And so, again, you can make predictions for the intracellular abundance of these metabolites, but I find it really curious that there is this essentially negative relationship between rate and fractionation, although it's the opposite trend that we see in sulfur isotopes, where you actually get larger fractionations in sulfur isotopes the slower the organisms are growing. So this is sort of, at this point, bringing it back together and asking the question of, just jumping ahead here, do sulfur isotopes in essentially the net catabolic products, sulfate and sulfide, as a function of net rate, and the hydrogen isotopes as sort of a proxy for the net anabolic products as a function of rate, can we actually combine these two isotope proxies and try to understand something or infer something about the way these microorganisms were growing, and essentially how effectively were they growing, how fast were they growing, how efficiently were they growing in natural systems, if we can go out and capture the sulfate and sulfide isotopic compositions, as well as their individual or their fatty acid isotopic compositions. I just want to make a quick plug before I move on to questions, in that I think this transhydrogenase in and of itself is actually going to be a really interesting transhydrogenase to study for not just in sulfate reducers, but all across anaerobes in particular. It's a very widely distributed transhydrogenase in anaerobes in particular, and it's a potentially very critical mechanism for energy conservation, this electron bifurcating transhydrogenase. And this is a really beautiful tree that some colleagues of ours published in a journal of biochemistry just recently, and I encourage you to check that out. But really, this transhydrogenase, I think, is there's more to it than just this hydrogen isotope story. I think it's really critical part of anaerobe energy metabolism in general. I mean, that's just some more data we can talk about another time. But yeah, so to sort of summarize the hydrogen isotope part of the story is that the fractionation between lipids and waters really nicely tracks growth rate and potentially intracellular redox rate. We're still working on a way to test this. And actually, if there's anybody out there today or in the future that watches this, if you have a good way of sort of quantifying and calculating net cellular redox, I would love to know, because that's something we're really hoping to sort of go after in future experiments. The transhydrogenase, the NFNAB, is probably critical in not just sulfate reducers, but anaerobes in general in setting their lipid isotopic composition. And yeah, I think this transhydrogenase in and of itself is a really intriguing enzyme to study, and there's actually a fair amount of biochemistry that's been done on it in methanogens and fermenters and other organisms as well. And so it's going to be really cool to see what comes out of the biochemical literature going forward. To sort of bring us all back, I've already said this, but really thinking about coupling these isotope systems, lipid hydrogen isotopes, potentially also lipid carbon isotopes, these anabolic isotopic systems with these catabolic isotope systems, so that essentially the electron acceptors and donors, you could think about doing this not just in sulfate reducers, but also in methanogens as well, or other anaerobes, or even other aerobes, where you can quantify essentially the net fractionation between their electron acceptor and then some proxy for their energy metabolism, and then some proxy for their carbon or their biomass building. And so we're actually moving on to do some of this work in archaea at the moment. This is what some of the work my master's students are starting to fire up. And I really like this joint approach of thinking about the microbiology and the geochemistry in a very experimental context, because that's essentially, I think, the way we're going to be able to better interpret signatures in both the modern sedimentary record as well as in the deep time rock record. And so before I take questions, I just wanted to highlight, this is all work that's been ongoing through my PhD and postdoc and ongoing in my lab, but we got a couple new folks in my lab just starting out, and they're doing some really cool work, just starting to look at the same proteomic and transcriptomic approaches and disproportionators versus sulfate reducers, and hopefully that'll be something we start to talk about in just a few months from now. And then carbon and hydrogen isotopes actually in aerobic methane production. That's actually a project that was inspired by a micro seminar by Paul Carini. And then essentially, yeah, continuing to differentiate these different anaerobic metabolisms on essentially multi-isotope scales. So thinking about hydrogen isotopes and methanogens and fermenters as well as sulfate reducers continuing. And then a really fun project in collaboration I have with folks, Seb Kopf and Ann Pearson and Eric Boyd, is looking at archaeal, the archaeal abundances and hydrogen isotopes in archaeal lipids, primarily from thermoacidophiles. And so hopefully, if I ever give another micro seminar, or you guys see me at Goldschmidt or AGU, this is what we'll be talking about in the near future. So with that, I'll shut up and take questions, and thank you all for listening in today.
All right, thanks so much. Well, that was awesome. That was great to hear what you've been thinking about. And so now Twitter, if you have questions, you can tweet us your questions. If you're here in the Hangout, feel free to chime in and unmute yourselves. And so I will moderate a few of those, but I was going to start and kick off with a question that I had, thinking about that cool figure that you showed, thinking about the electron scavenging with your slow growers. Mm-hmm. Just sort of wondering what you think, like what's your brainstorming about some of the mechanisms and pressures that they're under, and why they're cranking some of those sort of more electron shuttles up when you're starving them, and what that tells us about, like, likely that most microbes are starving most of the time out there in sediments, right? You think?
Well, yeah, no, that's in many ways, that's essentially one of the fundamental questions of how do we translate this very laboratory-based work, completely laboratory-based work, to, say, a natural system or even a well-studied system like your berry system. Is if we can go in and actually test that hypothesis, that everything, most things are starving most of the time. It seems like a reasonable statement, but I mean, I guess, what is the data that supports that? I'm sure there is some, and I'm just not coming up with it off the top of my head. But can we actually go in and look for, in a system that's well studied, like your pinkberry system or another simplified ecological system, simplified, right? Relative to a marine sediment, or I don't know, a wastewater reactor, and actually go in and look at, over either a naturally induced change in redox or electron donor availability, or like a diurnal cycle, or a human induced one, say something you do experimentally. Do we see the same crank up or crank down in electron acquisition machinery relative to oxidant acquisition machinery? And so to generalize, yeah, I think you can sort of extend that well beyond the sulfate reducer and just say, what is being cranked up under electron availability, or free energy availability versus essentially free energy starvation? And then, is that consistent with what we would predict? Are there cool new things that come out of that we wouldn't have predicted? Some of what we saw in the proteomic data is that, as per usual, in an expression experiment, you see all these hypotheticals cranking up in the slow organism. And so I just went back through that before this talk, and so now I'm like, oh great, what are all of these? Do we have any clue? Can we go sort of dive in from maybe somebody that's better at bioinformatics than me, and go in and sort of ask the question, do we know what any of these are? But then in terms of what we do know, a lot of these promoters and suppressors, that might actually tell us something. Before the talk, Lizzie and I were talking briefly about sulfur disproportionation, and this is sort of one thing that always bothers me is, what, if anything, is a sulfur disproportionator? They all just look like sulfate reducers, metabolic or enzymatically, or excuse me, genomically. But there are certain organisms that cannot be sulfate reducers that have all the machinery to do it. And so is there a way we can go in and essentially repeat these types of experiments on the proteomic scale to understand, is this just a matter of differential regulation, or is there something more to it than that? So that's still a very culture-based answer, but I think then applying that to maybe Lizzie's pinkberry system or other environmental systems where you're not totally sure what's going on, but you have a lot of other sort of maybe genomic or proteomic data and a lot of geochemical data that can sort of inform you what the net process is. I went off on a big tangent there. I don't know if that's your question.
Yeah, that totally did. All right, let's see if we've got questions from other people. Anybody in the Hangout want to chime in with some questions while I look at Twitter?
I've got one. So this is a really, I really enjoyed this, Will. Like, it's a very provocative talk in terms of energetics and how organisms are dealing with that, and then how it's being reflected in isotopic records. So I wanted to, especially the beginning got me thinking about this when you're talking about the total amount of oxidant available in a given form. Mm-hmm. And then your comments just now about how the organisms are, sort of responding to these. Do you think that the amount of electrons that can be moved from one molecular state to another matters, or is it really just about the concentration of the molecule itself? So like if an organism's not reducing sulfate all the way to sulfide, but it's reducing it to some other intermediate, is it capable, like, are we dealing with a situation where the organism can sense down to the amount of electron transfer, or is it just really about molecular concentrations?
That's a really good question. I should say that's a good question, and there's a lot of nuance to that question. So I have to think about that. So let me think about this. So I think it's essentially, it's always going to be reflected as basically the activity of the electron donor acceptor, or actually the ratio of the activities. So concentration, activity, yeah. I'm using them interchangeably, but I don't mean to. And then the organism, or actually the enzyme at this case, is ability to acquire, or the affinity of that enzyme, or that, you know, however you want to calculate that, either at the organismal scale, where we think of it as sort of a Ks, or at the enzymatic scale, where we think of it as more of a Km. And so it's essentially the activity or the concentration multiplied by the affinity of that organism or that enzyme, and that's really what's going to sort of set essentially the delta G of available for that reaction. But then actually, I think Doug LaRoe out of USC pointed this out really recently in a set of papers, is that even calculating the delta G, the free energy available in a reaction, that's all good. That's all well and good. But it's actually the rate then at which that reaction proceeds is what's going to allow you to calculate the power available in that reaction. Literally, how many watts can these organisms get out of, you know, a net set of reactions, or if you want to keep it simpler, just, you know, sulfite plus 6 electrons. But so it's not only just the affinity of the organism and the enzymes, the relative abundance of the different substrates, but it's actually that's going to set the delta G, and then it's the ability with which the organism cranks those through the reaction that sets the amount of free energy that they actually get out of the reaction. I don't know if that answered the question.
Yeah, I think I think I might be just being a little naive about the overall way that an organism is going to sense these things, because, you know, obviously it's going to have different enzymes for different electron donors, most likely.
Right. Sorry, I forgot.
was sort of thinking more along the lines of the, you know, your organic carbon experiments, as opposed to maybe like a, you know, inorganic oxidant, but, you know, something that where you have multiple different TCA cycle intermediates, and you're presumably expressing all the same enzymes to do this, but you can obviously get more electrons out of one intermediate than another. Is there, I don't know, so is fractionation being, I guess this isn't a well-formed idea yet, but I was just starting to think about maybe, instead of thinking about it from a molar standpoint from a molecule, but think more about it from a molar standpoint of the number of available electrons, and whether the organism can make decisions biochemically based on that.
Well, this is an interesting, I guess, couple questions, because so way back when I was reading some of Rolf Tauer's early work, when I was an early PhD student, and still trying to wrap my head around it. I'm still trying to wrap my head around it. But this idea of like vectorial electron transport, for example, and you know, basically the fact, and sort of this idea of electron bifurcation now, actually, that's sort of the newer and cooler idea in many ways, is that you can essentially, you know, drive certain reactions forward by, you know, essentially dragging them for, you know, energy requiring reactions with energy yielding reactions. Just, you know, net, that's just biochemistry, is what I like to say. But so I always sort of oversimplify it in my mind, and I'm wondering if that's problematic when I say it doesn't matter if you have 12 electron donors or 1 electron donor, because you can always sort of write out the reaction, or, you know, write out the stoichiometry of the 12 electron donor reaction, and then it's just going to be the net of that, the sum of all those reactions versus the, you know, the lactate, which is what I always use, because I work with the sulfovibrio vulgaris. But if you apply this into a natural system where you potentially have multiple electron donors, different organisms can only access a certain subset of those. You're going to have many different, not only you know, sort of possible reactions going on, but a lot of possible different redox free energies available, and potentially a lot of different rates going on simultaneously in the same, even maybe horizon. And so this, I think, is where then it becomes a really interesting question of, do we actually care, or at what level do we care about that nuance? Because if at the end of the day we're just measuring sulfate and sulfide dissolved in the sediment column or the water column, ultimately that's just reflecting the net of all of those different sort of those different organisms or those different subpopulations. But if for whatever reasons you do care and you want to ask questions about the ecology as you maybe go down with depth, then perhaps it becomes really important to know who's eating what, how fast, and, you know, what's their ability to use lactate or acetate or benzoate or something like that. I don't know if I just sidestepped your question or not.
No, I appreciate that. Yeah, I think this is just the beginning of a conversation, maybe
for sure.
time, but yeah, it's just the point is it's very you got me thinking about a bunch of random things that may develop later. So
for sure, yeah.
Thank you for that.
You have my email address. Yeah, it's applying the sort of the biochem, like the single enzyme scale where my biochemical colleagues work, or a few enzymes together, isotope effects across that, and then plugging that into actually a metabolic model where you're not only thinking about those reactions happening, but different amounts of those different enzymes, and then reactants and products and rates and fluxes, and then actually plugging that into not just the cell, but a population where every cell is probably going at a slightly different rate. And actually, one really cool observation from this guy, Seth Kopp, is that if you grow your population faster or slower, you get more homogeneous sort of data of consistent organisms growing all about the same rate. Sorry, this is a normal distribution of organisms growing at about the same rate when they're fast at the single cell level, but when they grow slower, or when you force them to grow slower in a chemostat, you get this more sort of like, sort of heterogeneity or, non-normal distribution at the individual cell level. And I think even observations like that just really make me scratch my head and be like, how do we apply this now to sort of not only my simplified metabolic models, but a natural system where you probably do have really, diverse populations growing at very different rates. And sort of back to Lizzie's question of, if everybody's starving all the time, what does an instantaneous rate measurement actually mean? And how do you interpret that in the context of a sediment profile, but then also thinking about, the production and preservation of these geochemical signals? Yeah. I hope that wasn't too much of a negative note.
I love starting to think about the heterogeneity at a cellular level amongst a presumably clonal population, just in terms of phenotype, right? So yeah, that's definitely the future for sure.
Yeah, and then applying these subcellular scale techniques that a lot of folks have used so brilliantly, and I think it's going to be really challenge. It's a challenge to me going forward, where I just take a whole population and say, you are basically one big cell. and actually starting to ask the question, well, okay, it might not be so simple, Will.
Mm-hmm. Mm-hmm. Yeah.
Cool. Got us all scratching our heads. Anyone else here in the Hangout have any questions for Will? I don't see anything here on Twitter, so if you got anything else?
I've got a quick question. This is Aaron. So I got kind of hung up on the plot that you showed that showed that the amount of DSRA produced between your fast and your slow cultures didn't vary at all.
Right.
Basically, and then I started trying to think about what that meant for sulfur isotope signatures, just in general, and does that sort of imply that there's no hope of being able to point at a sulfur isotope fractionation and being like, yes, that's a biogenic isotope signature, or hopefully I missed something there.
Yeah, no, and I know I dove into that really fast. My transitions were not so strong today. But yeah, so I think the answer is there is hope to interpreting sulfur isotopic signatures as biogenic or non-biogenic. But I think you're going to need at least another independent constraint to some extent. And then it's going to depend on the magnitude of the isotope effect. And then And so I'd say, if you're seeing sulfur isotope effects between sulfate and sulfide that are on the order of what we think of as equilibrium, so about 70 per mil, it's hard to imagine a circumstance outside of the laboratory, I should say, where you get such a large isotope effect, because that sulfate molecule is incredibly hard to break that sulfur oxygen bond at low temperature, meaning sub 150 Celsius, no weird metal catalyst or something like that. And so if you do see those really large isotope fractionations between a sulfate and a sulfide end member, or a barite and a pyrite, in sort of syn sedimentary, that I think you can, I would hedge that you can interpret that as biogenic, or more likely biogenic. Once you get into the smaller range in fractionation though, and I don't know, say you're on Mars or something like that, where it's not clear that you've had sulfate reduction necessarily. If you're into those smaller range in isotope space, sulfur isotope space, I think it's challenging to interpret that as biogenic. Did that address your question at all?
Yeah, thank you. That's great. Oh, I also wanted to make a plea for the anaerobic fungi. I think early on you said only bacteria and archaea are anaerobes. No,
I appreciate that, and I've actually had that in the past. So I intentionally sort of oversimplify, especially when I'm talking to pure geochemists. No offense to my geochemical colleagues, but I absolutely agree that fungi are probably doing some weird, incredibly cool anaerobic, energy metabolism that, beyond just yeast, which is of course important.
Yeah, and making beer.
I was gonna say, and beyond just actually disproportionation is essentially just inorganic sulfur fermentation, and so we have to look to the yeast literature if we really want to understand ethanol fermentation.
I wish Dave was here because I want him to chime in on say whether or not the fungi are doing anything at all without their bacterial endosymbionts.
Oh.
Well, so this would be interesting. Yeah, microanaerobic environments within fungi or something like that, within sediments.
And their mitochondria.
Yes. Are there intramitochondrial bacteria? No, that's tangential.
All right. So I think unless anyone has any other questions, Will is not on Twitter, but I'm working on strong-arming on there, so you will soon be able to pester him yourselves on Twitter.
I believe I have no choice at this point.
But if you have questions for Will as follow-up, you can check out his website, which has the coolest domain name I've ever seen. What is it? It's like geochemistry.rocks?
Geobio.rocks, and yes, that is a real domain.
So awesome. Anyway, you can contact Will directly. You can tweet us at MicroSeminar. Yeah, so we're looking forward to seeing you all in 2018. We've got a great lineup of folks. We'll get exact dates for our first January meeting out to you all soon. If you want to be in the lineup, we're hoping to do 1 to 2 microseminars per month in 2018. So don't feel like it's all filled up. There's room for you. So contact either me or Cameron or Dave if you're interested in giving microseminar next year. And with that, we will sign off. And thank you all. Thank you, Will, again, for doing this.
Thanks all so much for having me. It's great.
Yeah. All right. So happy New Year to everyone, and we will see you in 2018.